uh, started. It's my pleasure today to introduce Tao Zhong. He's a postdoc here in NC State, and we are very happy to hear your talk, Tao. Take it away.
Okay, thank you, Ruben. And hello, everyone. I'm Tao. I'm from Peter and Ruben's lab actually. So today I will talk about something about the genetic control of disease resistance in maize. So first, let's have a look at this form first. So this form shows the top 10 most destructive diseases in the soaring United States. So among of this, it's from 2022 to 2023, and among of these diseases, and there are some of these diseases we are interested in, like the gray leaf spot, the northern cone leaf blight, and the southern leaf blight. And we're also interested in some in the stock rot. And we found some stock rot diseases in here in the list, like the Asherah stock rot and the Gibberella stock rot. And I think the best way to control the disease resistance, I think, to control the disease, I think, is using the resistant QTLs or genes. And in order to identify the QTLs and genes, we can use the linkage analysis and also association analysis. And we can also, based on the gene's function reported in the other plant species, and we can identify the gene's function in maize too. And then when we have these genes or the QTLs for the resistance, we can use the traditional marker-assisted selection and to improve the maize embryonic resistance. And we can also use the genetic modification, or we can use the genome editing to improve the resistance. And today I will talk about two genes associated with the maize disease resistance. The first one is a MAPK kinase 45, and we identified this gene through the GWAS analysis. And we found this gene can improve the resistance to southern leaf blight, the northern leaf blight, and also gray leaf spot. And we also found another gene. This gene is a chitinase NAC, and we found this gene is associated with stock rot resistance in maize. And we tested two kinds of stock rot, Gibberella stock rot and Asherah stock rot. And for the first one, for the first gene, Dr. Yang Qin, she used the Goodman diversity panel. And also, we identified the SLB phenotype from 5 environments. And here, this picture shows the distribution of the SLB phenotype. And we see it looks like a normal distribution. So, and it means this data is good for the genetic analysis. And through the GWAS analysis, we identified like 10 candidate genes for SLB. Because Qin's lab works on some of these genes, so I just focused on this gene. This is a nitrogen activity protein kinase 45. After we have this candidate gene, we analyze this gene's protein sequence. And here, this is our candidate gene. And we found this gene is a kinase, and it's contained a MAP3K-like kinase domain. And we found this gene belong to the RAF subfamily. So we downloaded the RAF subfamily proteins from the maize genome, and we also compared these proteins with the other reported RAF proteins. And we found our candidate gene, there is another copy in the maize genome. It's named MAPK kinase 44. And we also found this gene is in the same cluster with the EDR1 in the rice and Arabidopsis. And the EDR1 means enhanced disease resistance. So it means our gene may be associated with disease resistance. So then we did a expression analysis using the SLB passenger to treat the W22, and we found our candidate gene 45. This gene can be induced by the passenger treatment, but the other copy, can't be induced. So we just focus on this candidate gene 45. And in order to identify this gene's function, we found a uniform mutant from the maize stock. And this mu insertion is in the UTR range here. And we self this mutant and we get the homozygous mutant. And we named it as W22MAP3K mu. And we tested this gene's expression level in the mutant and wild type. And we found, you see here, the expression level of this gene in the mutant is lower than in the wild type. And then we, using the wild type and mutant, we identify the SLB phenotype in the field. And we found the mutant is like more susceptible than the wild type. And we also identify this mutant and wild type phenotype in the growth chamber. And we found that, you see here, the lysine area is like, there are more lysines than the wild type. Oh, sorry. The wild type, the mutant has more lysines compared to the wild type. And we also identified the fungal biomass in the mutant and wild type. You see here, in the mutant, they have more fungal than in the wild type. And because we learn in the uniform mu insertion length, in the other locations in the genome may contain the other insertions. So we want to confirm this. We want to confirm this mu insertion is associated with the SLB resistant SLB phenotype. So we hybrid the mutant with W22 and several times get the F34 family length. And then we use two pair of primers to identify the genotype. The F and R is around the mu insertion, and the TRR6 is on the mu insertion. So if F and R there is no bonds, and the other pair of primers has a bond, it means this is a mutant, homozygous mutant. And if F and R has a bond, but the other pair of primers has no result, it means this is a wild type. And if both of these two pair of primers has bonds, it means this is a heterozygous. We identified the lysine area, and then we convert this data together based on the genotype. You see here, the wild type has this lysine areas compared to the mutant, so it means the wild type is more resistant. So this data suggested that the mu insertion is co-segregated. Oh, sorry, the SLB resistance co-segregate with the mu insertion. And then we also constructed an overexpression vector of this gene. And we have two independent transgenic events. And this is the first one. And we found the transgenic overexpression length, the expression level of this gene is just a little bit higher than the non-transgenic materials. And the second transgenic event, the expression level is much higher. And we identified this transgenic materials phenotype, SLB phenotype in the field in 2 different years. And for the first transgenic event, we found the positive, the transgenic length, the resistance is just a little bit, there's no difference, looks like. But the second one, you see they have some difference. You can see from the picture, the transgenic length resistance is higher than the non-transgenic materials. So overexpressing this gene can enhance the SLB resistance. So because we want to test whether overexpressing this gene could complement the SLB susceptible phenotype of the mutant. So we did two hybrid. We hybrid the homozygous mutant with the transgenetic receptor B104. And we also hybrid this homozygous mutant with heterozygous wild type, heterozygous overexpression line. And so for the first hybrid, they only have one genotype. It's non-transgenetics, and it's contained the heterozygous mu insertion. And for the second hybrid, they have two genotypes. One is non-transgenetics, the other one is transgenetics. And all of this, both these two genotypes contained the mu insertion. And then we identified this plant's SLB phenotype. Here, for this transgenic events, you see the transgenic maize has a higher resistance than the non-transgenic materials. And both these two transgenic events, they plant with the overexpression gene is more resistant than the plant hybrid with the receptor. So based on this result, we confirmed this gene, MAPKK45, is associated with SLB resistance. So then we want to confirm, or we want to figure out how this gene works. Because this gene named as MAPKK45, so we think maybe this gene is involved in the MAPK cascade pathway. And this picture shows the MAPK cascade in plant immunity. And here, this is the MAPK cascade. And the MAPK cascade can reserve the immunity signaling from like the receptor like cytoplasmic kinase. And then the phosphorylation level of this three kinds of kinase will upregulated, and then transducted the immunity signaling to the into the nuclear, so the transcription factors, and then improve the expression level of the immunity-related genes. And in order to confirm our gene is involved in the pathway, we using this antibody to identify the phosphorylation level of MPK3 and 6. And we also selected some SA and GA related genes, and we identified those genes expression level in the overexpression lines and mutant. Here, we use the overexpression lines, and this is the transgenic, this is the non-transgenic. We're using the SLB passenger to treat these two materials at different time point. And here we can see the passenger treat can induce the phosphorylation level of MPK3 and 6 here. But when we compare the phosphorylation level between these two genotypes, it looks like there is no difference. We also do this experiment by using the wild type and mutant. You see here it's the same. The passenger trait can induce the phosphorylation level of MPK3 and 6, but between these two genotypes, there is no difference. So based on this result, we can say this passenger can induce the MAPK3 and 6 activity, but overexpressing this gene or like lock down this gene's expression level will not affect the phosphorylation level of MPK3 and 6. And in the overexpression lines, we selected the three SA-related immunity gene, the NPR1, PR1, and also PR5. And we also selected the three GA-related genes, AOC, AOS, and LOX1. And we tested those genes expression level in the overexpression lines and also the non-transgenic materials. And we found there's no difference between these two genotypes. And we also do this experiment by using the mutant and wild type with the passenger treatment or without the passenger treatment. And for the 6 immunity-related genes with the passenger treatment can induce the expression level of these genes. You see here, this NPR1 is like pretty higher than the plant without the passenger treatment. But when we compared the expression level of these genes between these two genotypes with the passenger treatment or without the passenger treatment, there is no difference. So based on these two experiments, we hypothesized maybe our gene is unlikely to function through the classical MAPK cascade pathway. But we want to know how this gene works, and we want to know how this gene works. and we found this gene can regulate the ROS accumulation. Because SLB is a fungal disease, so we use the chitin. This is the chitin hygrosol. This is one kind of chitin to treat the transgenic OA expression nice and also the non-transgenic materials. And here we found by the chitin treatment in the OA expression nice, they have more ROS accumulation than the non-transgenic materials. And in the W22 and Y type, in the Y type and in the mutant, we see here in the Y type, they also has more ROS accumulation than in the mutant. So it means this gene can regulate the chitin-induced immunity response. And we also use the passenger to treat the materials, the overexpression nice and also mutant by using the DAB staining to test the ROS accumulation. And here, this is the transgenic materials. You see, it looks like they have more ROS accumulation than in the non-transgenic materials. And in the W22 with the passenger treatment, they have much more ROS accumulation than in the mutant. So this result suggested our gene can regulate the ROS accumulation. Then we want to know how this gene regulates the ROS accumulation. And we learn during the maize immunity response, the activity, the RBOS, can mediate the production of ROS. So in the maize genome, they have 6 RBOH genes. And we found 4 of them has like a normal expression level, and 3 of these 4 genes, 3 of these 4 can be induced by the SLB treatment. And like the RBH1, RBH3, and RBH4, the expression level can be induced by the SLB treatment. So we tested the expression level of these RBH genes in the overexpression lines. Here you can see this is the transgenic overexpression line, and this blue is the non-transgenic materials. You see for RBH1 and RBH4, the expression level is higher in the transgenic overexpression lines compared to the non-transgenic materials. And we also do this experiment in the mutant and Y type. And without the passenger treatment, the expression level of RBH3 and RBH4 in these 2 genotypes is similarly, but when we use the passenger treatment, the expression level is upregulated, but they both upregulated, but the expression level is higher in the Y type than in the mutant. So we think maybe this gene can modulate ROS accumulation, maybe in part at least, by regulating the expression of RBHS and then affecting the SLB resistance. Because we learn the ROS is a general resistance component, and ROS is also a immunity signaling. So we think maybe this gene can influence the other foliar disease resistance. So in the field, we using the mutant and the Y type, we identify the NLB phenotype, and we see here the mutant is more susceptible than the Y type. And we also use the F34 family lines. You see here, the mutant is more susceptible than the Y type. And we also tested as GLS resistance. The result is similarly with the NLB. And here is the summary of this gene. And by using the GWAS analysis, we identified 10 candidate genes associated with the SLB resistance in maize. And by using the mutant and the overexpression lines, we found this gene is positively regulates the SLB resistance. And we found this gene is not functioning through the classical MAPK cascade. And we found this gene can enhance RS production and maybe show the upregulation of the RBOH genes. And we found this gene can conform round spectral resistance to three kinds of familial disease: NLB, SLB, and GLS. Okay, here comes to the second gene. This gene is a chitinous neck gene, and we found this gene associated with stalk root resistance. And in maize, it has been reported mutant of this gene will affect the stalk strength and cellulose content. And also in cotton, the authors found overexpression this ZMCTL1's homolog gene GHCTL1 can enhance the vesiculum weight, and this is a this is a vesicular disease. So we hypothesize maybe this gene play a role in resistance to vesicular diseases. So we tested the stalk root. And in our lab, we have this gene's mutant, and the mu insertion is here, is also in the UTR ranger. And we identified this is the homozygous, here this is a homozygous mutant, and this is the wild type. We identified this CTL1's expression level in this two genotype. We found in the mutant, the expression level is much lower than in the wild type. And then in last summer, we tested the gibberella stalk root. Here you see the wild type is more resistant than the mutant for the gibberella stalk root. And for the astroclose stalk root, we have similarly result. You see the mutant is more susceptible than the wild type. So it's based on this result, we see mutant of this gene reduce resistance to stalk root. And as I mentioned before, for the uniformness, maybe it's contain other insertions in the other location in the genome. So we hybrid this now with this mutant with W22, and we select the homozygous wild type. Here is the capital CTL1 means the homozygous wild type, and the lowercase CTL1 means the homozygous mutant from the F34 family lines. And we also identified the gibberella stalk root. Here you see it's in the mutant, it's susceptible just a little bit. I think this result is not significantly between these two genotypes. And for the astroclose stalk root, you see it's the mutant. The mutant is more susceptible than the wild type. So I think based on this result, knock down the expression level of this gene can reduce the plant resistance to stalk root. And we also have this gene's overexpression nice. We have two transgenic events, and you see the expression level is much higher than the non-transgenic materials. But in the field, we found the overexpression nice, the plant hit is a little bit lower than the non-transgenic materials. And we tested the overexpression nice gibberella stalk root. Here you see the phenotype is great. And we can say the transgenic materials is more resistant than the non-transgenic materials. And we also tested the astroclose stalk. Here, stalk root, here you can see the transgenic materials is more resistant than the non-transgenic materials. So overexpressing this gene can enhance resistance to stalk root. And we also try to get the knockout materials of this gene. But we found here this picture shows in the greenhouse. In the greenhouse, we found knockout this gene, the plant we plant all of the plants planted at the same time. But you see the knockout plants, it's not grows normal, and they have some weird phenotype. And this picture is taken by yesterday. And you see it's also can't grow well. So we think maybe knockout this gene will cause the wilting phenotype in the leaves and severely impair the plant's development. And then want to know how this gene like function in the stop root resistance. Because this gene is a chitinase-like protein, so firstly, we want to examine whether it affects chitin-induced immunity or not. So we use the chitohexose. This is one kind of chitin to test the ROS accumulation. First, we do it in the overexpression nice. This is the transgenic overexpression line. This is the non-transgenic proteareas. And we see the ROS accumulation is similarly between these two genotypes. And we also do this experiment in the mutant. You see, between these two genotypes, this is the mutant, this is the wild type. There is also no difference. So it means this chitinase-like gene, CTL1, is not involved in chitin-induced immunity pathway. Because this is a chitinase, so I want to test whether this gene, whether this gene could like inhibit the growth of the passenger or not. So I express this gene in vitro in the egg, and I contacted this gene with the GST tape. Here, this is an purification of protein. Here, this is the GST. This is the protein with GST tape, and also confirm the result by using the Western blotting. Then I added some proteins in the medium, and then I spread the spores of these two kinds of insulate on the PDA medium, and then I put a filter paper on the medium, and then added different kinds of like proteins on the filter paper. Here I added some GST, and here is my protein, and this part is milk, and I also added some fungicide as a positive control. You see, for these two kinds of insulate, these two kinds of insulate can both induce the S-rocodose stop root. And here you see between the GST and our protein, there is no difference. So it means this gene will not suppress the passenger growth in vitro. And then we tested some marker gene, immunity-related marker genes expression level in the overexpression nice. And we found the expression level of NPR1, PR5, and ALS in the overexpression, in the transgenic overexpression nice. The expression level is higher than in the non-transgenic materials. So overexpressing this gene can influence the expression level of defense-related genes. But I still work on this gene, and I try to figure out how this gene works. I'm still working on. And here is a summary of this gene. And we found this gene can positively regulate resistance to stock root. And this protein, CTL1, will not affect chitin-induced immunity response. And because the knockout plants will can't grow, so we think maybe this gene is essential for maize growth and development. And we found this gene can't suppress the passenger growth in vitro. And here is something of my plan or my next step experiment. And because this is a chitinase-like gene, so I may want to test the chitinase activity. And we plan to do some iron-seq using the CRISPR-Cas9 knockout plants, because it's like you see the knockout this gene will severely influence the growth. And we also want to test the quantify the cell wall component content by using the knockout and overexpression plants. And maybe because we found some of the immunity marker genes expression level is upregulated, so maybe this gene will influence the plant hormone levels, and maybe I plan to measure the sugar content in the overexpression nice or in the knockout nice. this is just my plan. And here is the acknowledgement. And I want to thank Peter and Jim for their guidance for this project. And thanks Ruben for hosting me. And also I want to thank Shannon and Greg for their help and especially for the field work and the works in the greenhouse. And thanks for Wenyu for the discussion and help. And Qin and Tiffany also involved in this project. That's all. Thank you. Hi, Ruben.
Thank you, Tao.
Yeah.
Cool. There is one question here by John on the chat. John, do you want to go ahead and ask the question yourself?
Yeah. I was actually in the middle of sort of typing up the the ZMCTL stuff actually seems like it's a pretty interesting topic since if it is developmental, one idea just to kind of pitch is if there are other labs that might be doing like in situs or like looking at like spatial expression in developing tissue, you might get a lot of context just kind of knowing like what tissue types, approximately like what developmental stage that ZMCTL is coming on at. You already kind of answered my first question about the MAP kinase, where it sounds like with the reactive oxygen burst, it's probably like a hypersensitive immune response, is at least part of the story. I was kind of wondering though, where a lot of these defense genes are also sort of integrated into development and morphogenesis. Uh, have you thought or like sort of tried to interrogate, like, could there be like histoanatomical variation? Could there be like cell wall modification? So could you have like both active and passive defense in the MAP kinase as well, possibly? Or is it just sort of like an active defense? Sorry, that was a lot. That, that was a long.
Thank you, John. I don't fully got your questions, but the only thing I can say is like I think the CTL1 is involved in the cell wall development, I think. So maybe this gene is involved in associated with the Stockholm resistance. And sorry, I didn't got your questions. Sorry, I'm so sorry.
Oh, well, so like a lot of these MAP kinase genes, they
Yeah,
both will regulate like immunity, and so that's how you'll sort of get like these reactive oxygen bursts. But also sometimes they'll be involved in like regulating like cell differentiation, cell division, and also sort of like building up the cell wall, which kind of has me thinking like you made a really convincing case that like it's clearly involved in like active defense, it seems to be. I guess what I was also asking is could you have like pleiotropy in the system and maybe it's also involved in like building passive defense by modifying how the tissue is also built up as well. Um,
yeah, that's good. Good suggestions. We didn't like focus on that part of this gene. We just tested the resistance phenotype. And also, I think this gene, this MAP KKK, will not influence the development, I think.
okay, thank you.
Thank you, John.
But at the whole plant level, you haven't seen any obvious differences in development, right, Tao?
Yeah.
Are there are there more questions for Tao?
Oh, hi, Tao. I have a
Hi.
question about the expression pattern of the GMCTL. Because
Uh-huh.
you found knock out these genes, the field type is fair weird, and you want to do the unseq in the lookout line. And
Yeah.
I want to know which sample or which tissue do you want? I think this is
Oh,
will affect the early development. Maybe you need do the ASIC in the some or any other tissue.
oh, that's yeah. I take the samples for ANSIC yesterday. I just use this tool here. I use this. I use the force internodals here, and also the same from the knockout lines. I take the sample yesterday.
Okay.
Do you think this
So,
development, this stage is okay?
oh, so I want to know how there have some difference of the Lotto number between the white type
Oh,
and
the
normal
Lambda
type.
number is the same. I count. I just count like we only have 4 homozygous knockout plants, and I take 3 of them for iron-sick, and I count the nodal number from the year root to the root. The number is the same between these 4 plants, the H244 and the 3 knockout plants. It's the same. The number is the same, but the nonce
Oh, so the
is different.
okay, the last okay. Okay, thank you.
Thank you.
Cool. Are there more questions for Tao? Well, if not, thanks a lot, Tao, and we'll see everyone next week. Have fun at PAC if you're at PAC, and if not, we'll see everyone next week. Bye.
Thank you, everybody.
Yeah.